Explants of dorsal spinal cord are dissected from rat embryos and cultured in a three-dimensional collagen gel in apposition to COS cells expressing a candidate signaling molecule. The ability of the signaling molecule to reorient axons growing within the dorsal spinal explant is assessed using immunohistochemistry.
Hi, I'm Virginia Hazen from the laboratory of Samantha Butler in the Department of Biological Sciences at the University of Southern California.
I'm Phan, also from the Butler lab.
And I'm Ken Yamauchi from the Butler lab.
Today we will show you a procedure for culturing dorsal spinal explants and aggregates of transfected COS cells in a collagen matrix.
We use this procedure in our laboratory to assay the ability of candidate signaling molecules to reorient commissural spinal neurons.
In this experiment, we'll be examining the ability of Bone Morphogenetic Protein, or BMP7, to act as a chemorepellent. Let's get started.
Prepare COS cell aggregates used to challenge the trajectory of commissural axons from COS cells transfected with the expression plasmid for the molecule of interest, here bone morphogenetic protein, or BMP7. Aspirate the medium from the dish and rinse the cells with 1 milliliter of 1x PBS. Remove the PBS and treat the cells with half a milliliter of enzyme-free cell dissociation medium for 15 minutes. Stop the reaction by adding 1 milliliter of medium with fetal bovine serum. Using a 1000 microliter pipette tip, triturate the cells until none remain on the surface of the culture dish. Transfer the cells to a 15 milliliter conical tube and pellet the cells for 2 minutes at 2000 RPM.